So our next presenter is going to be Xin Liu. Xin actually is perhaps as well known across both buildings as anyone is going to be, because Xin worked for a long time at Dutton Park, where she worked with my father, Mike Roberts, who does similar sort of stuff to us, but more at a basic pharmacology and applied pharmacology level rather than a clinical level. But Xin has a capability in pharmacometrics, which she'll tell you about, the modelling aspect of maximising the value of data which is collected in pharmacokinetic studies. The reason that we wanted to highlight this specific study is, again, it's another example of a really important PK/PD study within a clinical trial, which is very nicely defined a finding which is going to lead to another clinical trial as well. So, Xin, please.
I'm going to present the project about the probenecid boosted oral antibiotic dosing in the SNAP trial. It's called PRO-SNAP. It's a sub-study of SNAP. SNAP is a novel randomized adaptive platform clinical trial. Evaluate a range of interventions with the aim to improve the outcomes for patients with Staphylococcus aureus bacteremia (SAB). The trial was led by researchers from Melbourne, but involved the sites from more than 10 countries all over the world. They recruit both pediatric and adult patients. The PRO-SNAP is a sub-study of SNAP, a prospective observational pharmacokinetic study. It performed in New Zealand, but our group contributed for the data analysis. It evaluated the potential of switching the prolonged IV antibiotic therapy to the oral, because the oral is more convenient, and then compare drug exposure between oral and the IV. Today's presentation will concentrate on the data analysis for cefazolin and cefalexin. The study include patients who was enrolled in the parent SNAP trial, and with the penicillin or methicillin susceptible SAB, age over 18 years old, and a patient currently on IV beta-lactam therapy, but with the potential to change to oral. Those patients with the unreliable gastrointestinal absorption or on renal replacement therapy was excluded. The patient will be collected 2 blood samples during one dosing interval. Patient could be sampled one dosing or several dosing intervals. And here's the characteristic for cefalexin cohort. Basically, the age was median age around 60 or older patient, and with the multiple ethnicity and with obese, the median weight around 90 kilo, and the BMI is over 30. And the dosing for the cefalexin include 1 gram giving orally 6 hours or 8 hourly, but with or without probenecid. And here's the characteristics for cefazolin cohort. They are basically similar characteristics. Some of the patients receive the cefalexin as well. For cefalozolin is being highlighted is the dosing regimen is variable, could be 2 grams 6 hourly, where the bolus or intermittent infusion, or the continuous infusion, or giving every 8 hourly. So once they collect the blood sample, we measured the unbound plasma concentrations for each drug, then generate this plasma concentration time profiles. Using this data, we performed population pharmacokinetic analysis. It allowed us to estimate the key PK parameters within the population. The key one we look at is volume distribution and the clearance. During the analysis, we found for cefazolin, the clearance was actually a little bit higher in patients with more Pacific background compared to the others. And also this renal function represented by eGFR have the significant effect on clearance. This was the same for the cefalexin as well. For cefalexin, another finding is with the probenecid co-administration, the clearance was reduced about 1/3. This means the drug exposure will be increased. And with the pop PK analysis, we also can estimate the inter-individual variability within the population. This was defined by this random effects, and the residual error tell us about the assay error. And these are just some goodness of phase plots and the visual predictive check plots. This allow us to evaluate whether the model could adequately describe the data and whether the variability within the population could adequately described. With this diagnostic plots, we are confident the model was valid. And then using this final model, we performed called Monte Carlo simulation. It simulates the concentration time profiles in 1,000 patients for the different dosing regimens. With this predefined the PK/PD target, we could estimate the probability of target attainment, which is the percentage of the patients simulated which can achieve this target. For optimal dosing, we aim for PTA over 90%. And this is the summary for the cefalexin. With the higher eGFR, the PTA was decreased. We test for two targets. First is 50% FT over MIC, aiming at 50% of the dosing interval. The drug concentration is above this minimal inhibitory concentration. And the high target is 100% FT over MIC. And you can see with the probenecid co-administration, it could increase the PTA. This one is the summary results for PTA for cefazolin. For this one, we separate analyzed for simulate for patient with more Pacific background and other patients because they have the different clearance. And also we test 3 different targets from the lower 50% FT over MIC to the highest 100% FT over 4 times MIC. And the finding from that is the PTA is lower in more Pacific patients because they have the higher clearance. And then the same with the increased eGFR, the PTA reduced means for those patients with the augmented renal clearance probably need to think for higher dose. And then the continuous infusion performs better than extended and intermittent infusion. The key finding is the standard 2 gram 8-hourly intermittent infusion could only achieve PTA over 90% for low target, but not for the higher target. Therefore, we propose a new treatment for the cefazolin dosing regimen using the optimized dosing, minimum 8 gram, maximum 12 gram, giving preferably as the continuous infusion within 24 hours. This can be tested within this SNAPS study to evaluate the patient outcome compared to the standard dosing. That's all. Thank you.
Thanks, Xin. And for the cefalexin project, what we found actually is that the exposures that you get with standard cefalexin dosing, so the oral component is actually quite reasonable. And so a new intervention within the trial is likely to be an early oral switch, earlier than what is currently the case with that cefalexin dose. So again, something which is really potentially clinically very meaningful. And of course, if you can convert patients to oral therapy earlier, they can be discharged from the hospital sooner. So very valuable.